A 6-month-old boy is brought to the clinic by his mother because of lethargy, irritability, vomiting, and tremors. He has a history of poor feeding with both breast milk and formula. On physical examination, the patient appears somnolent and a flapping tremor is observed. Papilloedema is seen on fundoscopic exam. Laboratory studies reveal an elevated orotic acid level in the blood and urine. Which of the following enzymes is most likely deficient in this child? Go ahead and pause the video and try the question yourself before I go into the solution. So before we tackle this vignette, it's important to understand what the urea cycle is. The urea cycle is a way for the body to take NH3 and convert it into urea because NH3 is toxic to the body and the brain. Urea is then excreted in the urine, and the way that this process is done is through what's called the urea cycle in the liver. The first step of the urea cycle is the combination of CO2, H2O, and ATP into carbamyl phosphate. And this enzyme, the enzyme that does this reaction, is carbamyl phosphate synthetase 1. I put a star next to carbamyl phosphate synthetase 1 because this enzyme is the rate-limiting step of the urea cycle. And it's important that we pay attention to this one because there's another carbamyl phosphate synthetase 2, which is taking place in pyrimidine synthesis that is not related to the urea cycle, and we'll go into that later. Carbamyl phosphate will then combine with ornithine via the enzyme ornithine transcarbamylase. So if you actually look at the enzyme name, so ornithine for ornithine and transcarbamylase, we have carbamyl phosphate. That's a way to kind of remember the reactants if you find yourself in a hunch, and this will create citrulline. Citrulline will then combine with aspartate to create argininosuccinate, and the enzyme that does this is argininosuccinate synthetase. So that's pretty nice. It's synthetasing argininosuccinate. Argininosuccinate will then be converted into arginine via the enzyme argininosuccinate lyase, and will give off fumarate, which is an intermediate in the Krebs cycle in this process. Arginine, so this is a very, very important thing to pay attention to. Arginine will then be converted into ornithine and urea via the enzyme arginase, which is a hydrolytic enzyme, and urea will then be excreted in the kidney. So now that we understand the urea cycle and the steps, there's one last thing that we have to pay attention to. So notice that the first two enzymes in the urea cycle, so enzyme 1 and enzyme 2, this is taking place in this kind of green square, and this green square signals that these reactions are occurring in the mitochondria, whereas the rest of the reactions are occurring in the cytosol. So that's something to pay attention to, is that ornithine transcarbamylase and carbamyl phosphate synthetase 1 are enzymatic reactions occurring in the mitochondria. So now that we understand the urea cycle, where does orotic acid come in? And that's going to be our next topic of discussion. So there's a process called de novo pyrimidine synthesis, and the entire point of this is to create pyrimidine bases for DNA synthesis and cellular division. De novo means from sort of coming from scratch, from nothing. And so the way that the first step of this, the first step of de novo pyrimidine synthesis is going to be catalyzed by the enzyme carbamyl phosphate synthetase 2. And this will combine glutamine, CO2, and ATP in order to create carbamyl phosphate. Carbamyl phosphate will then be converted into orotic acid, and the enzyme that actually does this is dihydroorotate dehydrogenase. So carbamyl phosphate and aspartate will combine to create orotic acid. And so if you have an elevation of carbamyl phosphate from a deficiency in the urea cycle, then from an inborn error of metabolism, such as an enzymatic defect in the urea cycle, then that carbamyl phosphate is going to be shunted to de novo pyrimidine synthesis, and you're going to have an increase in the amount of orotic acid produced. And that is what we're seeing in the laboratory findings. Okay, so going back to the vignette, a 6-month-old boy is brought to the clinic by his mother because of lethargy, irritability, vomiting, and tremors. He has a history of poor feeding with both breast milk and formula. So here, in the first two sentences, there's quite a bit of information that we can deduce. So we have lethargy, irritability, vomiting, and so this is suggestive of maybe malnutrition, but the tremors are very consistent with neurological defects. And the poor feeding with both breast milk and formula. This is very common in diseases such as inborn error of metabolism, lysosomal storage diseases, glycogen storage diseases. So that sort of narrows down our differential. The patient appearing somnolent and flapping tremors, more evidence of maybe malnutrition or neurologic defects. Papilledema, elevated intracranial pressure, more evidence of neurological defects. But really the most important part in terms of narrowing down our differential is the elevation of orotic acid level in the blood and urine. So which of the following enzymes is most likely deficient in this child? So let's sort of run through the answer choices. So the most likely enzymatic defect and what this patient is experiencing is ornithine transcarbamylase, or OTC deficiency. So the correct answer is E. And the reason why is because ornithine transcarbamylase, or OTC, converts the conversion of ornithine and carbamyl phosphate into citrulline. And so if this enzyme is defective, then there's not as much carbamyl phosphate being converted into citrulline. So that carbamyl phosphate is going to be shunted into de novo pyrimidine synthesis and cause elevation in orotic acid. And since there is elevated ammonia, that's the hyperammonemia, which is causing the neurological defects. So let's run through the answer choices. So arginase. Arginase, this is a really interesting answer choice because arginase converts arginine into urea and ornithine. So if you don't have enough urea, then ornithine transcarbamylase, if you don't have enough arginase, then urea and ornithine are not able to be created. So the enzyme ornithine transcarbamylase does not have as much substrate to be acting on. And so what ends up happening is that because there is not enough ornithine to be combined with carbamyl phosphate gets shunted into de novo pyrimidine synthesis and also would cause an elevation of orotic acid. And you would experience hyperammonemia and elevation of orotic acid. But the reason why this answer is incorrect is because what is most likely? Defect in arginase is not as likely as an ornithine transcarbamylase deficiency. So that is the flaw in answer A. B, branched chain alpha keto acid dehydrogenase deficiency. So this is what we see in what's called maple syrup urine disease, and we would also have neurological manifestations here. But the problem with this answer choice is that wouldn't explain the elevation of orotic acid in the blood and urine. Additionally, kind of a clue for this is sweet or syrupy smelling urine. Carbamyl phosphate synthetase 1. So a defect in carbamyl phosphate synthetase 1, that wouldn't really make sense because carbamyl phosphate getting shunted into pyrimidine synthesis is what's creating orotic acid. So C doesn't make sense. D, medium chain acyl-CoA dehydrogenase deficiency, so MCAD deficiency. This is a problem in metabolizing medium chain lipids, and this during the carnitine cycle, and this enzyme catalyzes the conversion of acyl-CoA into acetyl-CoA, but there really isn't any evidence of a lipid metabolism disorder happening in this vignette, and that wouldn't explain the elevation in orotic acid. F, uridine monophosphate synthetase disorder. So this is also a problem in de novo pyrimidine synthesis, and this also causes elevations in orotic acid. But a characteristic of this defect is megaloblastic anemia. So, and the reason why is because when there's not enough pyrimidine bases being produced, then that inhibits the erythropoiesis occurring in the bone marrow. And since there's no evidence of megaloblastic anemia, that's required to diagnose UMP synthetase deficiency. And so, by process of elimination, ornithine transcarbamylase has to be the right answer.